Streptomycin is a type of broad spectrum antibiotics which has been discovered after penicillin. The limitations of penicillin were it was a narrow spectrum antibiotic, and later on people or researcher came to know regarding the source of antibiotic, that is microorganisms. So they started searching for different types of antibiotics from microorganisms, and the second antibiotic discovered was streptomycin, produced from actinomycetes group of bacteria, that is streptomyces. Streptomyces is a type of actinomycete that produce aerial mycelium, and it also produces spores. So this feature is similar to fungi, but actinomycetes are prokaryotic organisms. The therapeutic use of streptomycin is like this. It is used in the treatment of infections caused by bacteria resistant to penicillin. So it is inhibitory to both gram positive as well as gram negative bacteria. Among gram negative, it is used for the treatment of infections caused by E. coli, Aerobacter aerogenes, Pseudomonas aeruginosa, Shigella, Serratia, etc. etc. So it is useful in the treatment of urinary tract infections, bacteremias, meningitis, and other infections caused by rare bacteria like Haemophilus influenza and tularemia. It has a slow positive effect on tuberculosis. So it is also effective against tuberculosis disease. It inhibits Bacillus species, Staphylococcus aureus, and also other type of bacteria. It is useful in the treatment, not only in the treatment of infections in animals, but it is also used in plant pathology in case of control of plant diseases caused by bacteria. For example, oily spot disease, which is caused in pomegranates caused by Xanthomonas axonopodis. So for the treatment of that disease, streptomycin is also used. In humans, prolonged use of streptomycin, a treatment at high doses, it may lead to side effects. So it may lead to neurological reactions, particularly difficulty in the maintenance of balance, partial hearing loss, etc. However, the reduction of streptomycin by chemical reaction to dihydrostreptomycin, it decreases this kind of neurotoxicity, and for this reason much of the streptomycin produced today is in the form of dihydrostreptomycin. It is available in the form of salt, or it is available in the form of hydrochloride, that is streptomycin hydrochloride. It is crystalline hydrochloride double salt with calcium chloride, and or it is also available as phosphate or sulfate, and dihydrostreptomycin is available as hydrochloride or sulfate. Now the major difference between streptomycin fermentation and penicillin fermentation is that streptomycin fermentation is not dependent on the use of precursors. That means the use of precursors does not increase the yield of streptomycin, which happens in case of penicillin. So streptomycin is a precursor independent fermentation. Let us briefly see the structure of streptomycin just for the sake of understanding. We are not supposed to remember the structure of this. This is just for our understanding. Streptomycin consists of three subunits called as streptidine, streptose, and N-methyl L-glucosamine. So structurally it is complex. This is hydrocarbon ring that makes streptidine unit. This is streptose and N-methyl L-glucosamine. So these three units join together to form streptomycin. Whereas in this region, if it is modified in case of formation of dihydrostreptomycin, here we can see that the here this CHO group is uh, substituted with hydrogen atoms and oxygen atoms. So that is called as dihydrostreptomycin. The properties of streptomycin are very important in order to understand or design the recovery procedure for streptomycin from fermentation broth. These properties are: it is a water soluble antibiotic, it is insoluble in solvents like acetone, chloroform, and ether. Therefore, these types of solvents can be used for precipitation of streptomycin during its separation from aqueous solution during recovery. It is readily absorbed on charcoal. So charcoal is the best material on which streptomycin is adsorbed, and therefore its elution is possible by using the dilute acid solutions and charcoal columns. Streptomycin is relatively stable as compared to penicillin, and that is why it is measured in terms of grams, micrograms, or milligrams. It is stable when stored at room temperature for 20 months, so it is quite stable compound. Less than 50% of activity of streptomycin was destroyed by heating at 100 degrees Celsius for 10 minutes. That means it is quite heat stable also. Strong alkaline solutions are destructive to streptomycin. It is highly resistant to action of enzymes and other biological agents. So these are the advantages. We have seen in case of penicillin, it is sensitive to penicillinase enzyme, which are produced by different contaminants, and contamination by penicillinase producer is a major threat to penicillin fermentation. However, in case of streptomycin, such type of problems does not arise. Now, the production of streptomycin. Commercial production of streptomycin is done by submerged fermentation process by using actinomycete species of highly mutated strain of Streptomyces griseus. Streptomyces griseus is a type of actinomycete that produce mycelium and spores. The raw materials used for the making of production medium are: the medium should contain the ingredients essential for the growth of Streptomyces griseus and for the production and accumulation of streptomycin. It is produced extracellularly. This is the general medium for production of streptomycin. It is glucose, peptone, meat extract, NaCl, and water. The final pH is 7.3 to 7.5. The nitrogen source may be replaced by distillers soluble soybean meal along with the addition of sodium chloride. The fermentation. The raw materials are mixed in a mixing tank at about 10 times concentration in which they used and pumped to a storage tank. From storage tank it is pumped into each of a series of 4 fermenters of increasing sizes as required. Water is added to dilute the nutrient solution to the desirable concentration, and then the medium is sterilized at 121 degrees Celsius, cooled down to the fermentation temperature, and then it is ready for the inoculation with starter culture. For the preparation of starter culture, that is seed culture, the spores from the stalk are transferred to sporulation medium. This sporulation medium, it provides enough sporulated growth, and it is done by using a liquid culture, liquid medium. These spores are then allowed to germinate in this liquid culture, and it is allowed to build up the mycelial inoculum, and then this mycelial inoculum may be transferred to the other higher capacity tanks, and this is how stepwise there will be development of mycelial inoculum. The fermentation temperature will be from 25 to 30 degrees Celsius. It is a submerged fermentation process with continuous aeration and agitation. The optimum pH for the production of streptomycin is between 7 to 8, with the highest rate of production in the range of 7.6 to 8. The fermentation period is 5 to 7 days, and the approximate yield from the industrial strain is 1200 micrograms per ml. This antibiotic is not destroyed by microorganisms as occurs in penicillin fermentation, although the other contaminants may infect the fermentation process and decrease the yield. Now, whatever the events that happens following inoculation of inoculum in production tank, it is called as the time course of production of streptomycin. So this course of production is divided into 3 phases. During the first phase, this first phase of fermentation, it lasts for initial 24 hours. So after the inoculation of mycelial inoculum into the production tank medium, initially there is rapid growth of mycelium. Streptomyces have a very strong proteolytic activity, means it has ability of production of proteases. So due to this, Streptomyces griseus, it releases ammonia. So the nitrogenous compounds present in the medium are soybean meal, and this soybean meal is degraded As a result of proteolysis, it will lead to the production of ammonia, and the carbon nutrients of soybean meal are utilized for the growth. So during the first phase is characterized by proteolysis and release of ammonia. The utilization of glucose during this phase is very slow. Slight streptomycin production may occur during this phase. Due to the release of ammonia, the pH of the medium increases to 7.5 or slightly higher. This is the optimum pH for the production of streptomycin. So during after this first phase, in the second phase, the streptomycin production starts at rapid rate. So it starts 24 hours onwards and it continues till the end of fermentation of 6 to 7 days of incubation. During this period, there is no mycelial growth. Whatever the ammonia which is present in the medium is utilized and the growth is rapidly withdrawn. Glucose is rapidly utilized. So rapid utilization of glucose and rapid synthesis of streptomycin are the characteristic of this second phase. The pH remains approximately between 7.5 to 8. The third phase. In the third phase, the sugar has been depleted from the medium. So sugar is being fully utilized. Then the streptomycin production stops, followed by the lysis of mycelia. As a result of lysis of mycelia of streptomyces, the ammonia which is present inside the mycelial cells is released and the pH may go beyond 8. However, the fermentation broth is harvested before the start of this phase. Then the recovery of streptomycin. The first step in the recovery of streptomycin is aimed at the separation of insoluble material. Since this fermentation product is extracellular, so obviously the first step for the recovery will be filtration. So at the completion of fermentation, the mycelium is separated from broth by filtration and the streptomycin is recovered by one of the several procedures. The choice of procedure depending on the particular industrial concern. So there are several filtration techniques that we have already learned. So in general, the drum filters are used for separation of mycelia. Second step, the aqueous broth which is separated following filtration is ready for the treatment with activated carbon or activated charcoal. Activated charcoal is then added to the clear to this fermentation broth, and the purpose of addition is clarification. This is done in tanks. So there are multiple tanks which are attached in a series. For example, 3 adsorption tanks. So in which the streptomycin and some of the impurities are extracted from broth by adsorption on the activated carbon, which then automatically fed to a pressure filter where the spent broth is separated out. So streptomycin has ability to get adsorbed on activated charcoal. So initially, after the addition of activated charcoal, the streptomycin will adsorb onto the charcoal. Then this charcoal is separated from the spent broth. So the impurities will leave with the aqueous spent broth, and streptomycin will remain adsorbed onto the charcoal. Then this charcoal, that is the adsorbate, this adsorbate charcoal, it is then washed with dilute alcohol. So what will happen, that whatever the impurities which are adsorbed on charcoal except streptomycin will be solubilized. So the purpose of addition of dilute alcohol is for the removal of impurities which are soluble in alcohol, whereas streptomycin will remain on activated charcoal. The next step is to separate streptomycin from activated carbon. That step is called as elution. In elution, streptomycin is eluted from the activated carbon by the addition of dilute solution of alcoholic hydrochloric acid by a two-stage countercurrent process. So addition of alcoholic hydrochloric acid will leads to the formation of a complex compound along with streptomycin, which is called as streptomycin trihydrochloride. So streptomycin will react with this hydrochloride and it will form trihydrochloride, and this will be released in the aqueous medium. That means it will leave the activated charcoal. So whatever the impurities are there, they will remain on activated charcoal, and streptomycin hydrochloride will be released in the medium, aqueous medium. Then this activated carbon is separated by filtration from the aqueous medium. Now this streptomycin trihydrochloride solution is ready for the next step. That step is called as concentration. So this streptomycin hydrochloride, which is called as the acid eluate, it is neutralized and then it is concentrated in evaporators. Means this solution will go to evaporators, where this solution will be exposed to 60 degrees Celsius or less. This will lead to the release or separation of water and alcohol. So there will be decrease in the water content and there will be release of alcohol content also. After concentration, the next step is solvent precipitation. Then this concentrate, which is obtained from earlier step, this concentrate obtained is then treated with acetone. So we have already learned that streptomycin is insoluble in acetone and therefore it can be used as a precipitating agent. So this concentrate is then treated by the addition of acetone. Addition of acetone will precipitate streptomycin hydrochloride, and due to its precipitation, its separation is possible. This mixture is then filtered through plate frame filter press, and the is filtrate is allowed to separate and it will go to solvent recovery unit. So here the precipitate is collected and whatever the filtrate is there, that filtrate will be only solvent, and that solvent will go to solvent recovery unit, and that means it will go to distillation plant. Then this precipitated streptomycin is then dissolved in alcohol. So dissolution in alcohol, precipitate will be dissolved, and then again it will be mixed with an adsorbing material that has been selected for the purpose of removing impurities, but not the streptomycin from the solution. So this is the additional step in which whatever the impurities which are there in alcohol, these impurities will adsorb on this material, leaving behind streptomycin. Then this mixture is filtered again so that the soluble impurities will be left out. Then this precipitate, or the precipitate which is dissolved in alcoholic solution, is now ready for the next step. In the next step, the alcoholic solution of calcium chloride. Now here the next step is addition of salt, calcium chloride. It is added to this alcoholic solution of streptomycin trihydrochloride, which is generated from the earlier step. So this will lead to the formation of a complex salt of streptomycin called as calcium chloride complex salt of streptomycin, and this crystalline complex is of high purity, that is 98% pure. During the final preparations under aseptic conditions, this crystalline complex is dissolved in pyrogen free water. It is then passed through bacteriological filters to get rid of microorganisms. Then after filtration, this preparation is dried in stainless steel trays from the frozen state under vacuum. Then after drying in vacuum, the powder which is obtained will go to a hammer mill. Hammer will be useful for the formation of homogeneous powdered formulation. Then this powder, it is weighed into, and it is then filled into sterile vials and packed. So the final preparation is obtained in the form of powder. The yield of streptomycin is approximately 1200. Sometimes the reported yield is 600 to 900 micrograms of streptomycin per ml in a shake flask fermentation. This is the flow sheet of streptomycin fermentation. Spore inoculum of Streptomyces griseus. It is inoculated into the inoculum medium that will generate the seed culture of mycelia. This mycelial inoculum is then inoculated into the production tank. The production tank has a temperature, or the temperature of operation is 25 to 30 degrees Celsius at pH 7. This is a submerged fermentation. Therefore, there will be continuous aeration and agitation. The fermentation will last for 5 to 7 days. The harvesting broth, that is the downstream processing, the first step in that is filtration. The mycelia will be separated. These mycelia will be washed several times with water, and the wash water will return to the filtrate just to avoid the loss of traces of streptomycin. Next step is the acidification of the broth. Acidification of broth is the additional step which will cause the precipitation of proteins which may be there in the fermentation broth. It is then filtered so that the precipitated proteins will be separated, and the filtrate, which is rich in streptomycin, is ready for the next step. Many of the industries make use of ion exchange resins. So there is use of cation exchanger column. So this use of ion exchange column chromatography we have learned in the last semester under the topic of downstream processing. Here a cation exchanger column is used for the separation of streptomycin. Elution with HCl is done, and the eluates are will then go to the evaporator. So evaporators are connected in series. That means it involves the drying at 60 degrees Celsius to get rid of alcohol and water. Then this concentrate is dissolved in methanol, followed by that filtration. Then the next step would be solvent precipitation, where acetone is used that will precipitate this streptomycin in the form of streptomycin hydrochloride. Then this streptomycin hydrochloride is dissolved in methanol. Then again it is filtered. After filtration, addition of calcium chloride as a precipitating agent that will convert streptomycin into streptomycin calcium chloride salt. It is then filter sterilized. The solution is filter sterilized, freeze dried, that is lyophilization, to obtain a crystalline fine powder, and then it is ready for the packaging in vials.